Good day, everyone. You are welcome to this online tutorials. The topic we'll be discussing today is an introduction to cell. And we want to review the concept of cell and the chemistry application to biological system. We also want to give you qualitative measure of how problems are existing within the cell, especially in a disease state. And so we start. You already knew that all organisms are built from cell. All animal tissue, including human tissue, are the collection of cells. And then we say that cell is a fundamental unit of life. That if cell dies, tissue dies. And if tissue dies, organs will die. And so the whole human system, human body will not be able to function. And as important as this statement is, we have come with 4 fundamental statements that will explain what we call cell theory. The statement number 1 says, cells are the building block of all organisms. All organisms are made up of cells. Without cell, there will be no organism. Statement number 2. All cells come from the division of pre-existing cells. Cells are the smallest unit that perform all vital physiological functions. As small as a cell is, it's able to maintain and function personally all its physiological function on its own. There are millions of cells in the body, but each of the cells function within itself, different from other cells. And so, in a cell, each cell maintains homeostasis at the cellular level. There is balancing within the cell. Then we say there is no living things on earth that is not made up of cell. The cell comes from other cells. At the cellular level, every individual cell maintains its homeostasis. And so the whole human being will be in homeostasis. But when the cell loses control of its own balancing with its environment, the human being will not be able to control itself. The human body will lose control. Introduction to cell continue. Animals, including humans, they have cells, and we say that their cells are larger in size. Nucleus is a site for informational component, and the site for this informational component in the cell is called chromatin. So in a nucleus, the region where this informational component is being carried out, processed and being carried out within the cell, we refer to it as chromatin. And the site where it is the respective site for generating ATP, converting food to energy, we refer to it as mitochondria. And in the site, and in this animal cell and in this nucleus, their reproduction involves mitosis and meiosis. Means this animal cell they carry out reproduction, and their reproduction involves mitosis and meiosis. When we talk about mitosis, we say that it's equal division of cell in which the cell divide to have 2 daughter cells. And it is a division of cell that takes place at the organs. Then when we talk about meiosis, Meiosis is a special division of a germ cell. When we talk about a germ cell, we talk about cell. It's a special division of a cell in which it results to 4 daughter cells. There are 2 types of cells. One, prokaryotic cell, which include bacteria and cyanobacteria. If you are talking about the prokaryotic cell, which include bacteria, we refer a good example of it is Escherichia coli. And the good example of the cyanobacteria is blue-green algae. You all know stagnant water or in a pond. What normally forms on top is what we refer as what? Blue-green algae is a good example of the cyanobacteria. And the prokaryotic cell, there is an organelles in the prokaryotic cell, but there is no membrane. Means the organelles is floating around the cytoplasm and it don't have membrane of its own. Means the only membrane you can see in the prokaryotic cell are the ones that is connecting with the extracellular fluid outside the cell. But in the cell and in the cytoplasm, the organelles are just floating around. There is nothing like membrane of the organelles in the cell. And their DNA is not complex with histone. In this lecture, I'll show you how DNA complex with histone look like. And the prokaryotic cell, the sexual reproduction does not involve mitosis or meiosis, just like with the one we have in animal cell, example in humans. In this prokaryotic cell, there is nothing like reproduction that involves mitosis or meiosis. And their genetic material is not enclosed by nuclear membrane, means their genetic material is not complex. They have genetic material, but it is not enclosed by nuclear membrane. 2. Prokaryotic cell. This includes protists, fungi, plants, and animals, including humans. I will keep discussing it as we go along this lecture. Cytology is a branch of cell biology. And cytology is the study of cell. And these are the kind of cell they study. They study cell. Cell referred to as germ cell or reproductive cell. They also study male sperm. They study female oocyte. These are the cells that develop into ova, what we refer to as egg in the female. And they also study somatic cell. Somatic cell are the division of the organs. And all body cell. All cells in the body, except the cell or germ cell or reproductive cell. All cells in the body, they study it. Now we move to cell membrane. Cell membrane. Extracellular fluid or interstitial fluid is a water medium that surrounds a cell. In the cell membrane, outside the cell, I will show you in the diagram, outside the cell there is something we call extracellular fluid or interstitial fluid. It's a water medium that surrounds the cell. And then that gives room for us to explain to you what we mean by plasma membrane. Plasma membrane separates cytoplasm from the extracellular fluid. Now we know we have a cell membrane. Outside the cell membrane, we have extracellular fluid. Then, if you move across the plasma membrane, you have the cytoplasm in the cell. So this plasma membrane is separating extracellular fluid from the cytoplasm in the cell, and is made up of plasma. That's what we refer to as plasma membrane. Then, what is the function of this plasma membrane? Number one function is physical isolation of cell from other cells in the same environment. They are able to do this physical isolation of cell from other cells in the same environment by they provide barrier to help the cell for what comes in and out. That is the barrier that they give. Barrier to help the cell for what comes in and out. Those things that comes in and out we refer to as ions and nutrients that comes in, or the waste material that is removed along with other product out of the cell to other location of the body. That is the physical isolation of the cell from other cell. So plasma membrane separate a cell from other cell. Even as much as the cell may be in the same environment, plasma membrane is acting as a barrier, isolating this cell from another cell. So this cell can function on its own, carry out homeostasis of its own, having a physiological function of its own from other cells. Number two of the function is regulation of exchange with the environment. What the material that they are exchanging with the environment is ions and nutrients. That the moment anything gets into the body, is in the body already. But plasma membrane have to regulate how it will get into the cell. And such material is ions and nutrients. It's also eliminate waste elimination, or all other products that have been produced or secreted, like the secretory vessel, out of other organelles in the body, in the cell, is able to eliminate it through this plasma membrane out of the cell. It's also helping the cell to maintain sensitivity to the environment. The cell becomes sensitive to the environment, to what is happening in the environment, through the chemical signals at the surface of the cell. Because at the surface of the cell, there is what we call receptors. And the function of the plasma membrane also help to provide structural support that anchor cells together to form tissues. Structural support. And it's also happen to anchor cells, means cells in the same environment come together and they are able to form tissues. And tissue in the same environment come together to form organs. An organ together to form systems, and they are interrelated. That is how we are able to have compartment within the bodies. We have come to a very important subheading which we refer to as membrane lipid. That is, this plasma membrane is made up of lipid, and in between the lipid there's something we call a phospholipid. This phospholipid have bilayers, and in this bilayer, in the phospholipid, it contains hydrophilic head. This hydrophilic head, it faces outward on both sides, means facing towards the cytoplasm in the cell, and also facing outward towards the extracellular fluid outside the cell. Then and there at the point of the cytoplasm there is water, and there at the point of the extracellular fluid which is outside the cell there is water. Means this hydrophilic head, they are water loving, so they move towards the water environment. But in hydrophobic tail is made up of two tails, not like that. That one moves towards inside the membrane, in which is not water loving. And also very important for us to know that this phosphobilayer acting as a barrier to ions and water. Barrier means water will not just flow from extracellular fluid and move into the cell across the plasma membrane anyhow. So because of the function of this phospholipid bilayer that I just explained, is able to provide that barriers to ions, nutrients, or waters, and that it works. This is a continuation of this lecture to explain better what cell membrane is. Listen carefully as we use this diagram to explain cell membrane. I explained earlier that cell membrane is made up of plasma, so we refer to it as plasma membrane. And it has a lot of functions. Functions such as physical isolation of cells from other cells in the same environment. And also provide barriers to ions and nutrients, even water that come into the cell. We also say that this cell membrane helps us to eliminate waste materials, secretory product out of the cell. Cell membrane also help us to maintain sensitivity to the environment of the cell through chemical receptors present at the surface of the cell. And these have made it possible for cell membrane to function properly. Then that takes us to what we said earlier about lipid membrane. This is the diagram of lipid membrane. I told you earlier that there is an extracellular fluid outside the cell and there is another cytoplasm inside the cell. And both of them, both extracellular and cytoplasm, they are water medium. And I also make you to understand that lipid membrane is made up of lipids and proteins. And you look at it here, look at globular protein, glycoproteins, look at cholesterol, look at glycolipid, look at peripheral proteins. All that we are talking about is protein and lipid, except this carbohydrate attached here. If not, lipid membrane is made up of protein and lipid. And in between them, I also mention cholesterol. Look at this place. Look at cholesterol here. That is in between the lipid membrane. And we say it's not water lovers. Now I want to explain to you now the phosphobilayers that is made up of hydrophilic heads. Look at it, head here, these are the heads, pointing towards outside the cell, which is extracellular fluid, and look at another one here. Look at another head, which is also hydrophilic head. These are head, and the head is pointing towards cytoplasm here. Then look at this head having two tails. Look at another head having two tails. This head having a tail pointing towards the cholesterol level here. Another head pointing towards the cholesterol level. That is what we call what? hydrophobic taste, having to take hydrophobic taste, which is moving far away from water medium. They are not water loving. And so we say this thing, it helps us to prevent barrier to ions, nutrients, and water that come into the cell. And it also helps us to eliminate waste and secretory product out of the cell to outside environment. So the functions of this lipid membrane is very, very important. And so now we will move further to explain what we refer to as cell. This is the structure of a cell, and at this region, this is what we refer to as extracellular fluid. This is what we refer as extracellular fluid, and inside there, this is what we refer as the cytoplasm. So here is extracellular fluid, here is cytoplasm. Inside this place, that is what we just destroy as what? Lipid membrane. And at this area, we refer to this one as cilia, at the surface of the cell, close to the pointing towards the extracellular fluid. At the plasma membrane, we have cilia. I will explain it to you later at the course of this lecture. If you can see this key here, we have, is this pink, we have yellow, and we have blue here. What we are demonstrating here is that what? All this blue inside this, they are membrane organelles, means they have membrane. Then all this one here that is having yellow color, they don't have organelle, they don't have membrane. They are just floating around the cytoplasm. While here we have our plasma membrane. And this plasma membrane contains what we call centros. Look at it here, centros. And inside this centros, around it, look at it, is in a centrosome. And the function of it is that what? It is essential for movement of chromosome during cell division. I told you that during cell divisions, which is at the division at the organs, mitosis, the existing cell divide to have two daughter cells. But if it is meiosis, the division, which is the division of the reproductive cell, to have four daughter cells. So means it involves essential movement of chromosomes during cell division. That is the main function of this centros in a centrosome. We continue with the introduction to cell, the cytoplasm. All material inside the cell are in the cytoplasm, which is outside of the nucleus. Look at this. This is our nucleus. This is our nucleus. Look at it. All this material here, which are inside the cell, they are in the cytoplasm, but is outside the nucleus. That's what we mean. Then, looking at now that takes us to cytosol. Cytosol is intracellular fluid, the water medium in the cytoplasm is the cytosol. So all the organelles in the cytoplasm attach itself to the cytosol. So the cytosol contains dissolved materials. The cytosol also contains nutrients, ions, proteins, and waste products. The cytosol also have high proteins and potassium levels. It has low carbohydrate, lipid, amino acid, and sodium levels. Now, organelles inside the cytosol. All the organelles inside the cytosomes, they all have specific function, as the name implies. So they have different function in the cytosol. Organelles within the cytoplasm. The organelles that do not have membranes, they are direct contact with the cytosol. Those organelles that are in direct contact with the cytosol, they include cytoskeleton, centrols, I've shown you centrols already, ribosomes, proteasomes, microvilli, cilia, and flagella. They are in the cytosol, direct contact with the cytosol, and no membrane. So if we say that organelle without membrane, these are the organelles. Then there are other organelles that have membrane. Apart from the plasma membrane, that is after the extracellular fluid before you come to the cytosol, this organelle that is in the cytoplasm also have its own membrane. And this membrane isolates it from the cytosol. And those organelles are the endoplasmic reticulum, the Golgi apparatus, lysosome, perosomes, and mitochondria. Organelle within the cytoplasm. One of these organelles within the cytoplasm, those ones that don't have membrane that are floating around the cytoplasm. One, the cytoskeleton. This cytoskeleton contains structural protein for shapes and strength. It provides structural support to the cell. It gives shape and strength to the cell. But it's not made up of bone. And we talk about structural protein. Those structural proteins that are able to allow him to give shape and strength to the cell. Proteins such as microfilament, intermediate filament, and microtubules. Microvilli. Microvilli increase surface area for more space for nutrient absorption. So for nutrient to be absorbed from the extracellular fluid outside the cell into the cell, microvilli need to increase the surface area for more space for nutrient absorption. It is also found in the digestive tracts of the small intestine. So the kind of cell and the location of the cell in the body that can possess this kind of protein, which we call microvilli, is in the digestive tract of the small intestine. And it also absorbs 90% of our dietary nutrients. Can you see why it is in the small intestine now? Because of nutrient absorptions. And this protein attach itself to the cytoskeletons. Now we are talking about the centrols. The centrole forms spindle apparatus during cell division. Mitosis, it pull apart chromosomes. Then the centrols is the centron that contain the centrols. The centrosome contain the centrosomes. Then cilia. Cilia is the slender extension of plasma membrane. This is the structure of a cell, but in this case we'll be demonstrating the cilia that I earlier showed you here. This is a cell and this is the cilia attached. This cilia that I'm showing you then, you see now that one of it now, as this thing as it may look like, is a microvilli or microvillus when it is if it is one. Look at this place, this thread-like structure called a microfilament. Then look at this region, we call it plasma membrane. In this lecture I'll be telling you about secretory vessel. So this is cytoskeleton, providing strength and structural support for the cells and its organelles. Both the cell itself and the organelles in the cell, they are able to interact between themselves through the help of this cytoskeletal component. So the cell is able to change shape, the organelles within the cell is able to change shape within the cell, even the cell itself is able to change shape, is able to maintain strength, and is able to maintain shape through the function of this cytoskeleton. Microvilli, if you are to use a microscope, electron microscope, to scan it properly, this is how it will appear. And this appearance you are seeing is the appearance of how it is in the cell. At this region we call it microfilaments. We are moving ahead. This is the structure of a cell. These are Golgi apparatus. This mitochondria. This is a endoplastic reticulum. This region that is smooth, we call it smooth endoplastic reticulum. This region where there is a spot attached to it, those spots we call it protein. Protein that is attaching to endoplastic reticulum. So we call it rough endoplastic reticulum. And they have their functions, and we will know the functions better as we go ahead. If you are to use special fluorescence labeling under the light micrograph, this microfilament, microvilli, look at how it is. You see the beauty of it. This is the beauty of it. As small as the cell structure is, all this as within the cell. That is why the cell is able to maintain its vital physiological function. The cell is able to maintain its homeostasis. The cell is able to carry out cell divisions. The cell is able to maintain its basic unit of life at the cost of all this arrangement. And then this one we have the microtubes. I've earlier shown you the centros. All this one, they are centros, but look at it. Inside that centros, we have what we call the microtubules inside that centrose, while the centrose is in the centrosome. That's the beauty of a cell. All I want you to understand is I get interested to understand how the cell is, how it looks like, and how it operates. Now we are taking one of the organelles now to discuss the functions and the compartment. Number one of the organelles we'll be taking is the ribosomes. Among all the others we have discussed, these ribosomes, they are organelles that synthesize proteins. These are the ribosomes. So if you are to know which part of the organelle that synthesize protein in the cell is called ribosomes. And the ribosomes compose of small and large ribosomal subunits. It also contains ribosomal RNA, which are made up of two parts. This ribosomal RNA, one of it is a free ribosome in the cytoplasm, which is used to manufacture protein that enter the cytosol directly. So this free ribosome is used to synthesize protein, but the protein that the free ribosome is synthesized, it enters the cytosol directly. Then we have fixed ribosomes, which are attached to endoplasmic reticulum. It is also used to manufacture proteins that enter endoplasmic reticulum for packaging and sending out of the cell. I also tell you that this manufactured proteins, this one is used to synthesize protein. The protein that is synthesized enters the cytosol directly, while this fixed ribosome also used to synthesize or manufacture protein, but this one enter endoplasmic reticulum for packaging and sending out of the cell. While the free ribosome enter the cytosol, this one now attach itself to endoplasmic reticulum. That's why we have smooth and rough. So the protein that is attached to this endoplasmic reticulum now, it will become rough endoplasmic reticulum. It's for packaging, which will be sent out of the cell. Another organelle is proteasomes. Proteasomes. These organelles contain enzymes, protease, enzymes that break down proteins. So now protein has been formed, but this protein need not to be broken down. We need an enzyme to do the functions. And this enzyme is a protease. All in these organelles of proteasomes. This protease also disables damaged protein. The protein has been damaged, is no longer useful for the body. It disables them and recycles them. Now we are talking about 5 types of membrane organelles. I've already mentioned it and I'll talk about it, that this one have membrane, which is 1. endoplasmic reticulum, 2. Golgi apparatus, 3. lysosome, 4. perosome, 5. mitochondria. So mention organelles that have membrane. These are the organelles. Mention and explain. You mention it and you explain it. If you are asked, mention organelles without membrane, the one without membrane, which are flowing around the cytoplasm, I've explained earlier, and I've also explained the function of it. Now, this structure will explain to you better. This is the structure of this structure is telling you the relationship between the rough and the smooth endoplasmic reticulum. So this is the structure of what? Endoplasmic reticulum. This structure is the structure of endoplasmic reticulum. Now here is to demonstrate to you how the things surround the nucleus. I will explain later how the nucleus is able to produce or secretory vessel or whatever is released, how it's able to move itself. I will explain all that later. But now look at how it arranges itself around the nucleus. Some have spot, some are plain. Like this one that is plain, we call it smooth endoplasmic reticulum. While this one that has spot attached to it, we call it rough endoplasmic reticulum. And from our earliest explanation, I've also want to make you to understand that endoplasmic reticulum contains storage chambers known as cystine. Endoplasmic reticulum contains storage chamber. There's a storage chamber called cystine, and the function of this endoplasmic reticulum is to synthesis of protein, synthesis of carbohydrate, synthesis of lipids. So I've told you before, I've shown you at this point that ribosomes is used to synthesize protein. Okay, but now on this point, we also now make you to understand that before we say the protein that is synthesized at this ribosome was sent itself to get itself attached to endoplasmic reticulum for packaging and sending out of the cell. That's what we say earlier. So there's no contradiction there. But at this point, we say now this endoplasmic reticulum now, it's also used to synthesize protein. First of all, protein is sent out of the ribosomes as it produced to attach itself to it, but it's also used to synthesize protein, carbohydrate, and lipids. So you see now, and it's also used for storage of synthesized molecules and material. Molecules and material that is synthesized used for storage is able to store. No wonder it's able to allow protein to attach itself to it. So it's not only protein that is attaching itself to it for storage, also molecules and materials are also stored. And also transport of material within endoplasmic reticulum. And also detoxification of drugs or toxins. So means here, I will not go into that in detail, but if we are talking about an enzyme that is used for detoxification of drugs, that enzyme will be located at this endoplasmic reticulum. Since at this point there is detoxification is only possible through the help of an enzyme. So it means here, where detoxification is taking place at this endoplasmic reticulum, there is an enzyme responsible, but I will not mention it. Let us not go into that for now. It will be located here. Now we have two parts of endoplasmic reticulum. We have smooth endoplasmic reticulum, which there is no attach of ribosomes, and at this point we also have rough endoplasmic reticulum. Look at it here. Rough endoplasmic reticulum, which is the surface is covered with what? Ribosomes. Like you can now see now, the ribosome that produce the protein that is produced on the package, it came here and it attached itself to these endoplasmic reticulums. Now, at these endoplasmic reticulums, just as what we told ourselves here, for synthesis of phospholipid and cholesterol for membrane. So at this endoplasmic reticulum, phospholipid and cholesterol for membrane is synthesized. Also steroid hormones for reproductive systems is also synthesized at these endoplasmic reticulums. This is under the smooth endoplasmic reticulums. Then under the rough endoplasmic reticulums, this is covered with ribosomes, is active in protein synthesis, and enclosed product in transport vesicle for delivering to Golgi apparatus. Can you see the line of it now? The line of it now is that here we are continuing I want you to listen attentively. We are discussing another organelles called Golgi apparatus or Golgi complex. Vesicles enter forming phase and exist maturing phase. And the function of this Golgi apparatus or Golgi complex are: 1. It modifies and packages secretions. And these secretions are such as hormones or enzymes for releasing from the cell. When the Golgi apparatus package these secretions, it also helps to modify it in the way that it will be released from the cell. And such secretions we mention hormones or enzymes. Number 2, at the cost of releasing out of the cell, some of these secretions is very, very important to the cell, for the cell to survive, and some is merely waste that need to be eliminated out of the cell. So some of these secretions, it helps to renew or modify the plasma membranes. You know that membranes is made up of lipids and protein, and some of these secretions is made up of proteins that will be released to the other target organs or vital organs of the body. So it means it also helps to renew. This protein can also help or renew or modify the plasma membrane. Number 3, it also helps to package special enzymes within the vesicles. Enzymes such as lysosome for use in the cytoplasm. And the Golgi also produce lysosome. So the Golgi's apparatus is not only helping to package the enzymes within the vesicle loop, but it's also part of the productions of this lysosome. Now the lysosome's enzymes, which is part of the enzyme produced by this Golgi apparatus. The function of these lysosome enzymes is that it's a powerful enzymes containing vesicle produced by Golgi apparatus. It's a very powerful enzyme. What makes it so powerful? Listen carefully now. The function of it is to destroy bacteria. That is why it is powerful. Means for bacteria to be destroyed within the cell, not to be able to break down the cell or destroy the cell, it has to be destroyed. The bacteria need to be killed, and it is done by this powerful enzyme that we call lysosome enzymes. It also helps to break down molecules and recycle damaged organelles. So the organelles that is damaged need to be recycled. And it also has another powerful function, which is autolysis. This autolysis is a self-destruction of damaged or inactive cells. The cells that have been damaged or due to injury that won't be able to survive again, the cell will automatically on its own commit suicide. It is powerful enzymes that help the cell to commit suicide in order not to affect other cells. And that is what we refer to as what? Autolysis. as a function of this lysosome enzyme. Lysosome break down lysosome membrane breakdowns. How will it be able to do this function of self-destruction of damaged cells? It by what? Breaking down the membranes. It also helps to release digestive enzymes. You know, I told you earlier that it's an enzyme. So the digestive enzyme that is released for digestion, it is out of this lysosome. Cell is also destroyed, which are part of the function I've explained, and also cellular materials are recycled. Now we want to explain secretory product that is produced in a nucleus at the cause of the information contained in the chromatin. This secretory product can be protein or waste that need to be laminated or any other important material within the cell that is produced. Now look at how it's able to attach itself to this rough endoplasmic reticulums and then able to repackage, look at it here, in a transport vesicle to the Golgi apparatus. You already know the function of the Golgi apparatus, and that is which we explain here, that it helps to what? Modify and package secretions. And now it is released out of the cell as the process of exocytosis. It is those waste that have been modified and repackaged. Some of them are protein that is necessary, vital to other parts of the body that need to be released. Some of them can be waste. Now it is eliminated out of the cell as the process of exocytosis. This one is explained better. You see, this is the lysosome. This is the transport vesicle. What is inside here, we are referring to it as the lysosome enzymes. And I mentioned maturity phase. Listen and look at it carefully. And then all that we are demonstrating here is how this important secretion is able to modify, repackage, and release out of the cell as a process of exocytosis. This is the, now we are on the Golgi apparatus again. I want you to understand something. And this is a diagram to illustrate what I've said earlier. This is the structure of the cell. At this space, this is endoplasmic reticulums, and this is the Golgi apparatus. Now, within the cell, but you know it is outside the nucleus. And after the protein has been produced, attaching itself to rough endoplasmic reticulum, it is packaged and released into Golgi apparatus. This is the secretory product. What is inside is a secretory product. This one is what we are seeing here. All this opening that you are seeing here, look at it here. They are transport vesicle, and now it is able to transport those material out of the cell and the process as the process of exocytosis. We are moving to explain further on another organelle called perosomes. Perosomes, they are small enzymes containing vesicles. They are produced by division of existing peroxisomes. Means it's the existing peroxisomes that divide to produce new peroxisomes. They break down organic compounds such as fatty acid. For heavy organic compound to be broken down is through these organelles called peroxisomes. And we are moving into another organelles which we refer to mitochondria, smooth outer membrane. I will show you in the diagram better. They are smooth outer membrane. Inner membrane has numerous food which are referred to as crystal. I will show you. Crystals surround fluid content, which is called a matrix. It takes chemical energy from food, which is form of glucose. So, and this glucose produces the energy that we call ATP. Okay, so the food you are eating, after the food has been absorbed from the blood system into the cell, it needs to generate ATP. So the food is converted to glucose, which is to produce ATP. ATP that will sustain the cell and sustain the entire bodies. So without ATP, there is no way human being will be able to breathe. Now we are moving to nucleus. They are largest organelles. The largest organelles in the cell is nucleus. The cell control center is the one that controls the whole cell. It's the cell control center. And that is where we talk about the informations of the genetic code, the genetic materials, and even for protein to be produced, synthesized. Everything happens at the cause of the DNA that is present in the nucleus. Here we have nuclear envelope. I will show you double membrane around the nucleus, perinuclear space, and then this communicate message into nuclear membrane. Now this is the function. Okay, I want you to listen carefully. This cell nucleus, now we want to discuss all that I've mentioned earlier now. That the content of the cell, we talk about nuclear matrix in the nucleoplasma, support filament, nucleoli, nuclear organelles. This nucleoli synthesize ribosomal RNA and assemble the ribosome subunit. It's also made up of RNA, enzymes, and histones. Nucleosomes, DNA coiled around the histones, and they are usually called into chromatin in non-dividing cell. Then this one we can explain the function better of the cell nucleus. The cell nucleus and the information storage in the nucleus. This is the major function of this nucleus in the cell. They are the information storage in the nucleus. This information storage in the nucleus are in form of a genetic code. And this genetic code, they are the chemical language of the DNA instruction. This code, this code is being used to give instructions. And the code is made up of sequence of bases, which we refer to as A, T, C, and G. And if we are talking about the triple codons of the DNA, out of this A, T, C, and G, at the end, you have 3 codes that will make a codon. And that's what we are talking about. Triplet codon means three of this code. And through this process that we're able to have the major 20 amino acid in the body. We continue. the function of nucleus in the cell. One of the functions of nucleus in the cell is information storage. Information storage. All the information that control the cell, down to the tissue, organs, and the whole system, it is stored in the nucleus in form of DNA. If the DNA is to replicate, it is stored in this place. The information is here. And if the DNA is to be transcribed to messenger RNA and also translate itself to proteins, everything is done in the nucleus. And it is done in form of a genetic code. In form of a special code. And this genetic code is a rules used by living cells to translate information coded within genetic material into proteins. So for protein to be formed, the instruction given in form of a rule using genetic code. And this genetic code is a chemical language of the DNA instruction. So the instructions that the DNA will release, if a protein is to be formed, it will come in form of a genetic code. And this genetic code is in a sequence of bases. And these are the bases: A, T, C, and G. If you are looking at it later, you begin to understand that this A is represent different amino acids that need to be formed, and different instructions are the result of this code. And if the total amino acid that will give rise to protein synthesis out of this nucleus is in form of a triplet codon. This triplet codons, the instruction is contained in a gene. It tells a cell on how to make protein. The information is contained in the gene. This triplet codon will tell a cell how to make protein. We say gene is DNA instruction for one protein and is the functional unit of heredities. We are going ahead. I have seen a lot about nucleus when I was mentioning all the materials and every other important thing around the nucleus. At this region that look like plasma, we refer to it as nucleoplasm. And you see another thing that look like a thread-like structure inside. We refer to it as a chromatin. So, if the information, genetic material, instructions, everything present in the DNA is present in the DNA and it is in the nucleus, at what location can we see it? The site is called as chromatin. And look at this, we refer to this one as nucleolus. And at this region we refer to as nuclear envelopes. Then you look at a spore here. Look at pore. Something that looks like pore. We call it, refer to as nuclear pore. And these are important structures of a nucleus. Now we have now single out one of these pore. Let's explain it better. Now look at it. If you're able to single out the pore to continue, nuclear pore, and that was the label I told you earlier, and something that looked like a thread-like that you are seeing here. A thread-like, look at it that you are seeing here. If you bring it out, we refer to it as a nuclear envelope, and in between we refer to it as perinuclear space. As we are moving ahead, listen carefully now. This is the nucleus in the cell, preparing for cell divisions. Look at how look at the appearance of it, where the nucleus is preparing for cell division. When the cell is preparing for cell divisions, the nucleus inside, look how it looks like. This is not preparing for division. Look how it appears. Now, at this point, when the cell is preparing for cell divisions, it come out, it form this formation you are seeing inside. This is the appearance of it as the visible chromosomes. And if we are to show the supracoid region, this supracoid region, you look at it here. But what I want you to understand here is the winding of this DNA around histone. I told you earlier that I will tell you what histone is all about. So you see this winding of this DNA. Look at this around protein. What is inside this thread-like structure that is winding itself around this region? What is there is protein. So histone is a basic protein. It's basically protein. And at this region of this winding around this protein, we refer to as nucleosomes. So now you want to know the structure of DNA. Look at how the thing is. Double helix. Look at how it's showing itself here now. So these are the beauties of what happened within the cell. And I want you to go over the class and understand what we are talking about. Thank you for listening.